Hello, everybody. Good morning to all the people here in the Western Hemisphere. Good evening to everybody who is back there in the east side of the wall. As we wait people to get logged in, I just want to thank and welcome everybody to the ATBC webinar on sociality and multispecies interaction among vertebrates. My name is Gabriel Colorado. I'm a professor at the National University of Colombia, back right now in Medellin, Colombia. It's 8 a.m. here, and I want to initially thank Alejandra Jaramillo from Universidad Militar Nueva Granada, who's going to be hosting us today as a ATBC counselor, and thanks for making this event possible.
Thank
Great.
you very much, Gabriel, and hope everything goes smoothly.
Yeah, I also wanted to thank Alejandra. She's the chair of the Capacity Building Committee at ATBC. So we'll try to explain why we think this is capacity building in a little bit. But I wanted to say hi, I'm Evan, and I'm over here in China, and it's good evening over here. Also, April 10th. Yeah. So, yeah, Gabriel, do you want to talk a little bit more about our format and what we expect?
Great. So thank you, Evan. So Evan and I were going to help out with the moderation of this webinar. We have three dynamic speakers, but before I introduce them, I want to thank all of the participants who are joining now, who have been waiting for this space. So as probably we all know, each speaker is going to speak for about 20 minutes. After the three talks are done, we are going to open up the space for questions, and we hope to spend about 30 minutes in the question and answer space. The whole session will be available on the ATBC YouTube channel soon after we're finishing the event. Please go ahead, Evan.
Great. So everyone, we're very thankful that you guys are here, or the participants listening. We want you to keep your questions. So think of a good question. It's going to come at the end of those three, right? And you're going to put them in the chat box. And then because there's a number of people here, so things don't get chaotic, Gabriel or I are going to try to pull stuff from that chat box and ask our speakers. So I think basically, please keep yourself on mute. And if you have any technical problems, something not working, give a private chat to Gabriel or me. You'll see us over on the listing of the people. Yeah, so this is kind of the standard format for ATBC webinars, but they started a little bit before this, right, Gabriel?
Yes, yes. So this is part of a webinar series. The ATBC started way back in 2000, just during pandemic, or before pandemic. And there was actually a bio-rate interaction webinar that was organized by Emilio Bruna and Lisa Comida. And that was related to plants and interaction with herbivores. We thought we would like to contrast that with a kind of different approach, a different subject, but also related to bio-rate interactions. In this case, we're gonna be hosting a webinar on interaction among vertebrates, animals, and mixed species animal aggregations. Yeah, Ben.
Yeah, and so our purpose here is to generate excitement because we think these are really, really cool phenomena. We hope you will agree. And we want you to get excitement rolling for ATBC 2025. It's going to be shout out to a conference. It's going to be in June 29th to July 4th in Oaxaca, which is a really awesome place. We hope you all can come. At the same time, we want to say, you know, it's hard, it's expensive to go to conferences, right? And we learned so much from the COVID pandemic about this kind of stuff. And so we were real believers in this kind of stuff. And we think that some of the society's mission, like outreach, like discussion to talk about stuff that we all are interested in, can be done in this kind of format. So that's why we consider this capacity building.
Okay, so yeah, that's our mission here. So we went back to a symposium that was done online on 2021, and then it was written up in a special issue of the philosophical transaction of the Royal Society in 2023, all about mixed species animal groups and aggregation. So we look for contributors To that volume, we study different kind of animals, in special birds, mammals, and fishes, so we can have kind of an understanding of the diversity of these groups, as well as looking for some similarities among them. So we also look for young dynamic speakers and researchers who are working in tropics and the subtropics. So with that, I would like to introduce our first speakers.
Okay, can I interrupt you there, Gabriel?
Okay.
Yeah, what's the order of our speakers?
We're going to start with birds.
Oh yeah, I like that. I like that. It's a good idea, huh?
Okay, so full disclosure here, Evan and I, we work with birds, right?
Yeah. And so the order is birds, B for birds, F for fish, M for mammals, right? Oops, I forgot that probably doesn't work for Spanish. One of our ideas had been to have Spanish subtitles. We didn't get that quite, but okay. Anyway.
Okay. So we'll let our audience get back to us, and then regardless of the order here, I'm really pleased and like to introduce Dr. Giselle Mangini. Giselle is a postdoctoral fellow at Laboratory of Bird Ecology and Animal Plant Interaction at Instituto de Ecologia Regional, IER, Universidad Nacional de Tucumán, Consejo Nacional de Investigaciones, Científicas y Técnicas in Argentina. Giselle works mainly in the Yungas forests in Argentina. She studied the dynamics of mixed species bird flocks and their ecological interactions. Passionate about understanding nature beyond isolated snapshots, her work explores how behavior, food resources, and environmental factors shape bird interactions. She's currently conducting behavioral and bioacoustic studies to understand how different species communicate, the roles of a species within mixed species flocks, and the implications of these social interactions for ecosystem function. Today, Giselle will talk to us about followers, leaders, and freeloaders, unraveling the dynamic ecology of mixed species bird flock. So Giselle, the floor is yours. Thank you again for being here with us today.
Hi, everybody. Okay, I'm going to put my presentation. Okay, share some. Share. And everybody seeing the presentation? See?
Yes.
Perfect.
Yes.
Well, okay. So, first of all, thank you, Gabriel, Evan, and Alejandra for making this possible. For me, it's really exciting. And in this presentation, I'm going to talk about part of my research on mixed species flocks of birds. But before we start, let me know if this like that. But before we start, I want to show you like this short video of different species that you can find in a mixed species flock, right? They are from different places, but I want to pay attention for the different type of behavior, the coloration. Sometimes we can find big woodpeckers, small woodpeckers, and sometimes we even can find house wrens within mixed species flocks. But what is a mixed species flocks of birds, right? So researchers has reached a consensus in which a mixed flocks is a group of different bird species that move together while foraging over a non-aggregated food resources. And the consensus is that individuals have to be together for more than 5 minutes and in close proximity between them, with a minimum of 3 participants from 2 different bird species. But sometimes we can find mixed flock of 100 individuals belonging to 30 different species. Some squirrels can join the mixed flock. So it's a really complex behavior, right? And the majority of the birds that join mixed flocks are insectivorous birds. So 60% or more of the species in a bird community may join a mixed flock, and this highlights how striking behavior for the bird species in a community mixed flock is, right? They are described for the whole world except in the Antarctica. And up to 13 mixed flock types have been described depending on the composition of the species, the habitat, and the behavior. So for example, we can find mixed flocks in open environment, in savanna environment, and in forest environment, even in the understory, in the canopy, or sometimes even a multi-level mixed flock that encompass the two levels. It has also been described that species in mixed flocks has roles. So for example, we have the sentinel role that is described as species or an individual that performs alarm calls against predators. There is also described the leader role that will be species that move in first position, nuclear species that contributes to the formation and or cohesion of the mixed flocks, and followers will be species that follow the leaders, and depending on different scenarios, they can take another role. So we can think that part of bird repertoire or foraging decisions include who to forage with, and sometimes they will decide to join a mixed flocks, right? But why will a bird join a mixed flock? Well, it has been proposed two main hypotheses that can work for all type of animal aggregations, that mainly an individual in a group diminish the chances of being predated and increase the chances of foraging more efficiently. But especially for mixed flocks of birds, there are some mechanisms that will be making these benefits possible. So especially for diminished chances of being predated, we have more individuals within a flock surveying the environment, so they have more chances to detect predators. At the same time, some species will have the sentinel role, although it has been described that some of them perform false alarm calls to cheat other individuals and steal the food from others. And the mobbing behavior, that is when species mob a potential predator, but sometimes because they get too close to the predator, they end up being eaten. And the different mechanisms related to how birds will be able to increase the foraging is, for one side, imagine a volume of the forest, right? Instead of one individual, you have a group of individuals that will be a mixed flock moving there. So just because they are more in numbers, they will scare more away more predators, more arthropods. And at the same time, each species has its own feeding technique to make arthropods more visible. So it's like a combination between numbers and feeding techniques that enhance visibility of arthropods for birds in mixed species flocks. At the same time, within a flock, individuals can watch successful foragers and mimic the foraging behavior. And at the same time, there is a collective knowledge of recently depleted areas, so birds can avoid surveying areas that has been already surveyed before and doesn't have any food. So in combination, all these mechanisms, for one side, will diminish the chances of being predated for a participant bird, and at the same time enhance the foraging. While they diminish the chances of being predated, this enables more time to invest in foraging for birds. So they are receiving like a double benefit in the foraging, or we can think that, right? So I start to wonder if we can think in mixed flocks as a coping mechanism for birds to overcome challenging situations, right? And when I start to think in what will be a challenging situation for birds, I realize that we have a lot of changing factors for birds that bird has to predict, and some of them change within a year and others change within a day. So the first thing that I did was to evaluate how the temperature, that is really straightforward to think about it in changes within a year and within a day, influence the flock formation. And if you see here, every season has a color, and you can find mixed flocks in every season. But as temperature increase, you have less chances to find a mixed flocks. And at the same time, bird responds to this decreasing temperature within a 24 hours temporal window. But I continue thinking whether other things that could be changing for birds, right? If you can see here, there is the same forest, different seasons, and you can see the striking differences in foliage cover. But at the same time, a lot of things are changing too. So we have some flowers blooming, some fruits getting ripe, breeding birds, and even the reproductive cycle of arthropods change along the year. But within a day, arthropods that are ectotherm depends on the temperature to move, right? So if it was a cold day, we have less mobility in the arthropods that is in comparison when it's a hot day that we have more mobility in arthropods. So I try to measure and quantify all this, and I end up proposing this hypothesis in which how birds perceive foraging opportunities and decide whether to join or not a mixed flock will depend on short and long-term changing factors. For one side, we have how many arthropods are in the environment, but at the same time, we have how hidden those arthropods are. And these hidden arthropods will depend on the type of arthropod that is in the environment. So let's say if it's a higher proportion of caterpillar or pupa species in comparison to butterfly species, we will have less visibility for birds when they look for arthropods, right? And at the same time, if the foliage cover is higher, arthropods will be more hidden. And in the same way, if there is a cold day, arthropods will be less mobile, being more hidden for birds to eat. So when I analyze all these variables together, I realized that birds decide to join mixed flock depending on a combination of variables that change in the long term and in the short term, right? Especially those variables that contribute to making the arthropods more difficult to find. So after this finding, I start to think about whether birds are actually obtaining benefits by joining mixed flocks, right? And if we think in the foraging efficiency and in the foraging behavior of birds, the morphology and habitat can give us some clues about what the bird is going to do. If we have a big woodpecker, we can expect that it forages on a big branch. If we have a small woodpecker, we can expect that it forages on a small branch, right? But sometimes birds can surprise us, right? This is called what is called plasticity or flexibility in birds' behavior. We can see hummingbird feeding on sap on trees, some granivorous bird feeding on excretion of insects, or even woodpecker feeding on fruits from cactus, right? And probably the behavior that bird has are related to different hypotheses or ideas. So from one side, we can expect that birds attempt to maximize the energy that they obtain per unit of search effort. But at the same time, they need to balance the nutrients that they are intaking. So each species has an hypothetical space required in which they try to consume different food items to achieve that hypothetical space. So for example, here in the highlighted yellow image, we have the same species, but it can eat caterpillars or it can eat fruit, right? And sometimes, why not, saltator can feed on leaves. And if we keep thinking about this foraging behavior, we have that the feeding can be understood like a combination between search for food and consume food for birds. So what I did was to spend a lot of years recording bird behavior, describing this behavior that bird has, analyzing this behavior. And what I found was that when a bird is inside a mixed flock, right, is being part of a mixed flock, both searches and consumption increase, right? So when a bird is inside a flock, it's only thinking about eating, it's only feeding, right? But the consumption here, you can see that it increased more than twice when compared to the search effort. So based on that, I start to think, okay, but we have different flocks participants. And if you remember from the beginning, I mentioned that species have different roles, right? So I start to wonder whether the leaders, the one that goes first, really want to be followed and share the resources that they found first, right? So I start to wonder who is benefiting the most within a mixed flock. The first thing that I need was to analyze who was the leader species in my case. So I start to read a lot of information, and I end up finding that, for example, in the Amazon, there is one species or one genus that is usually described as leader, nuclear, and sentinel species. They are the Anschreik from the Tamnomane genus, right? And here you can see a video of the species. It's a female, is calling in the middle, and you can see all the other flock participants in the back, right? It has a really steady behavior when it's in flock. But if I keep reading, I went, so for example, to the Atlantic forest that is in Brazil and Argentina. There is a red crown antanager and golden crown warbler that are described as leader or nuclear for the understory mixed flocks in the Atlantic forest. And in this Atlantic forest, you can also find the Cinereus anschrank that we have just seen, but here it does not fulfill any particular role or is not so important, right? So I start to realize that birds can change the behavior depending on the environment. And I want to show you here a video of the golden crown warbler, so the nuclear species for the Atlantic forest. And you can see that it's a more dynamic species in comparison to the anschrank. And another example for Taiwan, it has been described as Ciper morrisonia as a nuclear species, right? And it's really interesting because this is a gregarious species. So instead of having one or two individuals per flocks, every time you have a flock, you have several individuals fulfilling or behaving as a nuclear species. So as you can see, it's really different depending on different environment, how this goes. So I realized that I needed to create a standardized way to characterize the flocking, the roles of the species in mixed flocks. So I started thinking whether I can compare the behavior of movement, the behavior of being a leader with a structural property in a network. And I started to analyze whether these criteria called neighborhood inclusion criteria, that what it does is summarize the interaction that a node has. So I created a huge network with all my mixed flock participants and compared it with the behavior that I observed on the field. And I found out that the frequency of leader behavior was associated to this structural property in the network. So I was able to tease apart who was a leader using a structural property of a network and comparing with the behavior of the species. But even more exciting, I found that 35 species behave as leader in my study area, that it was the Yungas forest at the east of the Andes, right? And even more interesting, I found that there is a hierarchy in this behavior. So we have this species is a gray Elaenia, right? If this species is not so abundant in my study area, but when it's present, it has the leader role. If this species is absent, this species takes the leader role. If this species is absent, this other species take the leader role. So there is a hierarchy. There are stronger and weaker leaders. And probably this is related to the foraging benefit that I end up finding, right? So what I found was that leaders are species that are naturally more active than followers. They perform more searches and more consumption than followers. But when they are inside mixed flocks, leader has 8% less search effort, but increase the consumption 22%. So for a leader, being in a flock is extremely benefiting. So contrary to what I expected, species want to be leaders in flock, or that is what I think, right? But if you remember, we also have like different other categories to classify species. We have some species that lives in the canopy and feed on insects, species that live in the understory and feed from seeds, and all these different species make use of mixed species flocks. So to give you some examples, we have this tropical parula that we can think is a foliage insectivore. When it's inside mixed flock, the consumption rate increases, the search rate remains the same, and they diminish the forage height. So they are being more efficient in the foraging when they are in flocks, but they are also changing part of the foraging behavior. We also have the example of Asayaka tanager. It's increasing the consumption rate, the search rate, and is foraging higher when it's inside mixed flock. And this is probably related to the fact that when this species is inside a mixed flock, it starts to eat mainly arthropods. So if you remember, we have a bird that consumes a fruit usually is more steady than a bird that has to follow and pursue arthropods, right? So that's why the behavior of this species is becoming more active. And an example that I really like is the case of the woodpeckers, that we can say that they are bark foragers, right? We have 4 species in my study area, and we can see, if you pay attention to the yellow arrow, when the bigger and the smaller woodpecker species in the study area are being benefited when they are inside flocks. And at the same time, all the woodpecker species in the area start to forage higher when they are inside mixed species flocks, but only 2 species start to forage higher in a significantly way, right? So I start to dig in this idea of what is going on with the woodpeckers. And I find that the average mixed species flock size in the area is 7 species. But when woodpeckers, but the average mixed flock with woodpecker has 12 species without counting the woodpecker, right? So the woodpeckers seem to prefer to join bigger mixed species flocks. And in 80% of the mixed flock that they have woodpeckers, I also found that there were another woodcreeper or bark forager species. So when I start to analyze the association preference of different woodpecker species, I found out that each species has its own preference to be in flocks with certain species. So for example, these species never interact with the biggest woodcreeper in the area, and this species that is epiculate, the smallest species of woodpecker in the area, it never interact with the biggest woodcreeper in the area, right? And something really interesting that I found was we have this narrow-billed woodcreeper, right? It was foraging on the ant larvae here in this hole. And immediately after it went down, a couple of these woodpecker species came, first started to forage the male and after the female. So what is going on here is that birds inside a mixed flock, I found that they are not monopolizing the food resources, right? We have that in the first place. And perhaps woodcreepers are enabling fast food for woodpeckers. Otherwise, woodpeckers need to find a place that has soft bark, dig a hole to be able to actually forage, right? But here, they are just following these woodcreepers that are finding more exposed food. So regardless of grouping, the foraging benefits that bird species obtain are probably related to a combination of behaviors, species-specific traits, and maybe specific circumstances that are going on in the environment. And finally, for you to, like the main conclusion and the takeaway message will be that mixed flocks are dynamic, right? They are part of the daily decision-making of the birds. And birds seem to join them as a coping mechanism to deal with difficult situations like bad weather or difficulty in finding food. And within the optimal foraging theory, birds are not merely balancing the energy that they intake, but rather minimizing uncertainty, right? So birds in a flock can balance the nutrient intake. They can switch from frugivorous to insectivorous diet. They can modify the foraging behavior, and sometimes leveraging the behavior of others to make faster and, why not, smarter foraging decisions. So whether by following, leading, or freeloading, birds in mixed flocks are solving what we can call a part of the ecological puzzle of foraging decision. Okay, thank you. I hope that everything went okay.
Yeah, great. Thanks so much.
Okay.
Now, I have many birdie questions on those woodpeckers I want to ask, but I'm not going to say them. I'm going to keep them, put them in the chat box, right, guys? Okay, because we're going to go on to our next speaker. And it's my honor to introduce Dr. Matt Henson. Matthew is a postdoctoral researcher working in Berlin, Germany at the IGB Leibniz Institute of Freshwater Ecology and Island Fisheries. His work focuses on the functions and mechanisms of group hunting in pelagic environments. He currently leads a DFG-funded project exploring the interactions between billfish and other predators, avian, mammalian, teleost, as they attack schools of small fish. Today, he's going to talk to us about multi-predator feeding aggregations in the pelagic. Please go ahead.
Cool, thank you very much. Let me share this. So thank you very much for the invitation, Eben and Gabriel, and for the ATBC for putting this on. I'm just going to close my video for the talk, and I'll put it back on again afterwards. So I usually like to start with a tour around the world. So this is the sardine run in South Africa. This is Norway. And this is off the coast of Western Australia. And this is one of my field sites in Baja, California. And what you're looking at is a school of sardine, often called a bait ball, being attacked by mahi-mahi and sea lion. Now, if we compare these images from around the world, there's an obvious pattern. There's an incredible diversity of predator phenotypes. They're all attacking within groups.
Yo no estoy en eso, entonces.
Sí, yo quedé aburrida con el otro.
Pues claro, se habló de su protocolo.
Could you mute yourself? Sorry, I can hear people.
Okay.
Cool, thank you, sorry. So where was I? Yeah, but there's a very similar prey phenotype. So these large schools of clupioid fish. Now, it's often argued that this pattern occurs due to the type of primary production. So essentially, there's a very large amount of single-celled phytoplankton, and this equates to an abundance of zooplankton for these small-bodied clupioids, which then grow fast and reproduce quickly, and then they become the dominant food source for this huge array of larger predators, including humans, of course, and these small forage fish actually comprise over a quarter of our own fishery. And so we can be pretty sure that it's of obvious ecological and economic importance. Now, these feeding aggregations have also been a mainstay of nature documentaries for decades, but actual scientific investigation to the behavior occurring at the aggregations has really lagged behind. For example, what we know about their terrestrial counterparts. So the first thing to highlight here is that in these terrestrial aggregations, the predators are aggregating around a carcass, which is obviously has very different properties to a school of fish. For starters, it's dead. It's essentially immobile, but it's also structurally very complex, composed of bones, muscles, organs, and skin. And now, once a predator makes the kill, usually of a relatively large prey item compared to the size of the predator, you controlling access to this valuable resource becomes a priority activity. And the between predator interactions are characterized really by these dominance hierarchies. Now, usually this means that larger or stronger predators win out first. However, weaker predators can overcome them by forming larger groups. And in fact, competition over the carcass is often listed as a factor explaining the evolution of group living in some of these predators. Now, the carcass's structural complexity means that species can focus on separate tissue types, and we know that the feeding activity of some predators can facilitate prey access for others. So, for example, some smaller birds need the skin to be opened up by these larger predators before they can actually feed. Now, these interactions have obvious effects on community dynamics. Just an example, they can result in intra-guild predation. Now, let's compare this prey type again to what we've seen in the pelagic. So here the prey are numerous. There isn't a single kill, but there's this continuous harvesting of the resource as the prey kind of avoids capture as a collective. Now, the prey are smaller than their predators, so their top speeds are slower, so they can't outrun the predators. But what they are more maneuverable. And the prey rely really heavily on collective defenses, where the sufficient transfer of information between schoolmates allows the individuals to react really quickly to threats, even those that they don't experience firsthand. Now, predators, therefore, have evolved counter strategies to counteract this. For example, herding or prey dispersion or between predator coordination. And kind of what we have here is something akin to a predator-prey arms race. So the prey is attempting to maintain this cohesion, and the predators are trying to disrupt that cohesion and capitalize on isolation. Now, considering these very different types of prey type between these terrestrial examples I showed you and these pelagic ones, there's no strong reason to assume their functions and mechanisms are really directly translatable. But really, to investigate this, we need a tractable pelagic system, which takes us to my study systems. So these are 2 field sites on either side of the Pacific Ocean. Every November and December, about 30 kilometers off the Pacific coast of Baja, California, these striped marlin aggregate to hunt sardine schools. And in October, a very similar predator, sailfish, aggregate in Kuala Lumpur in Malaysia to hunt a similar prey type. Now, in both these locations, alongside these 2 species of billfish, we get a huge diversity of other predators. So we have different teleos like skipjack or needlefish, mahi-mahi, different pinnipeds, cetaceans, and then various bird species, so frigates, terns, and pelicans, which amongst themselves have very different phenotypes as well. Now, secondarily, so really there's kind of dominant questions that I'm trying to answer since these very fundamental beginning early on questions. Now, firstly, we want to know, what is the nature of the interactions between these predators? Are the interactions purely competitive? Maybe some predators facilitate prey captures for other predators. Perhaps the interactions are even mutualistic, with both predator species benefiting from each other's presence. And we also want to know what role phenotypic constraints play. So how does the morphology or the physiology of the predators determine the nature of these interactions? Now, just like in terrestrial systems, these really fundamental questions are really the first steps towards a better understanding of the species foraging ecology, which leads on to community dynamics, and then ultimately an understanding of ecosystem resilience. Now, the problem, however, is that to answer these fundamental questions, we ideally want data that goes beyond which predators are present or absent. However, it's just been simply very difficult up until now to record things like capture and attack rates and to quantify the spatial temporal position of these animals. But thanks to some advancements in underwater and aerial video and tracking technology, this is now becoming more possible. So what we try and do is we try and record the hunt from 2 perspectives, from the air and from underwater simultaneously. It kind of looks like this. Now I've just labeled the same predators in either viewpoint. And this provides a sufficient level of detail to measure the attack and capture rates of individually identified predators, as well as the spatial temporal positioning of both predator and prey from underwater and from the air, which critically can be converted into these real-world coordinates. Now, this last point is actually quite important because it allows us to relate the movement of the predators and prey to attack and capture data. So here you can see a trajectory of a prey school, and the X and Y axis are in meters. Now, all these little black X's along the trajectory, these are predator attacks. And with this kind of data, you can produce some important insights. So for example, and this is something that we can remember for later in the talk, these faster moving prey schools, when they're chased at high speeds, they produce higher numbers of isolated and therefore easily captured prey. You can see an isolated one being caught there. Now, there are, of course, many possible interactions occurring within this diversity of predators, but for today's talk, we'll mention 3. So striped marlin and Californian sea lions, sailfish and bridled terns, and striped marlin and mahi-mahi. Now, to remind you, we're curious about the nature of the interactions between the predators and also how the morphology or physiology might play a role. So let's start with marlin and sea lions. Well, our observations showed us that while marlin were often hunting alone or in single species groups, the sea lions never were. They were always found in the presence of marlin. So they were always in mixed species aggregations. Now, by using underwater video, we could create for the very first time a data set of attacks and captures for 2 predator species attacking the same prey school. And you can see that here on the right, where each blue dot is a marlin attack and each red dot a sea lion attack. And it's just across time in the bottom, on the axis on the bottom. Now, what we found is that in mixed groups, so the attack interval of striped marlin and the capture interval of striped marlin was higher. And this meant that the competitive presence of sea lions interfered with the hunting behavior of striped marlin. Now, we combine this with the fact that we never saw sea lions hunting by themselves. We induced that the sea lions kleptoparasitize this relationship and that we have evidence of interference competition. Now, this data, however, was all from underwater footage, and we're yet to analyze how the interactions between striped marlin and sea lions, they might alter when the prey school is being chased around the ocean. And it's possible that here, sea lions help to fragment the prey school and may therefore actually create a benefit to striped marlin, so at least in certain contexts. Now, regarding sailfish and bridle terns, PhD student Max Licht was curious about these relationships between underwater predators and aerial ones. And he collated 3 hypotheses from the literature about why feeding aggregations of these predator groups are so common. The first is that underwater predators can drive prey to the surface where it becomes available to birds, at least those that can't dive. Second is that scraps from underwater predation activity are fed on by birds. And the third is that underwater predation activity breaks down the cohesion in the prey school, making captures easier for birds. Now, by using drone video again, Max could assess the shape of the prey school as it was attacked by sailfish, and then he could link this to the timing of the predator attacks. So that's what it looked. This is like a still shot from the drone. That's what it looks like. And what Max found is that as the prey school expands during a sailfish attack and takes multiple seconds to recover to its original shape and size, and this green asterisk here is when the sailfish attacks. He also found that the terns attack when the prey school has expanded. And this red asterisk here is when the tern attacks. So lastly, what he did is he showed that the majority of tern attacks, like these blue dots, all occur in the seconds following the sailfish attack. Now, so in summary, what Max was able to show is that sailfish provide a one-sided benefit to terns by lowering the cohesion of prey schools, and that a decrease in the cohesion of the prey school can trigger subsequent attacks. And this is evidence of facilitation or maybe even commensalism. So far we have example evidence of competition where sea lions reduce the captures of striped marlin. We have evidence of facilitation where terns time their attacks to occur after sailfish have dispersed the school. But what about mutualisms? Is there any evidence of that? So I wanted to start this last multi-predator interaction by looking at some environmental data. Now, the red dotted line in each of these yearly graphs of sea surface temperature represents 25 degrees Celsius. Now, note please in 2023, how the sea surface temperature rose well above this line in this October, November, December period. That's when we do our field work. And these pie charts here, they just show the proportion of different predators observed at the aggregations. Now, they're dominated every year by striped marlin in blue and frigate birds in orange. But notice in 2023, that striped marlin slice of the pie shrinks, and we get a new species dominating, and this represents mahi-mahi, which were both very common and also found in very large groups in 2023. Now, we look at the physiology of mahi-mahi and compare it with striped marlin. The literature tells us, at least, that mahi prefer and perform better at warmer temperatures. They really like it around 27, 28 degrees. And we hypothesize that the warmer sea surface temperature either pushed or pulled them further north than they normally are found at this time of year, which brought them to our field site and to hunt alongside striped marlin. Now, this is a video of them hunting together. There are a few captures in this video, actually. I'll just let you watch it. The mahi are extremely efficient hunters. They catch almost a sardine almost every second attack, which is pretty remarkable. Okay, so in 2023, recorded 40 hunts, and we observed 3 different teleos predators, so striped marlin, mahi-mahi, and skipjack tuna. And if we look at the occurrence of these different teleo species across the 40 hunts, over half of the hunts involved only mahi-mahi without a different teleos present. And they were often found in these huge schools hunting with sea lions. And it's really too difficult to record very accurate attack and capture rates when they're at these numbers. But curiously, in about a third of the hunts, they were alongside striped marlin. And when the striped marlin, their overall numbers of predators was much smaller, and therefore we could assess the nature of their interactions by measuring the number of attacks and the number of captures by the different predators. So when mahi were present, striped marlin attack rate dropped quite considerably, but curiously, the capture rate was actually mostly maintained. Now, this is unexpected because normally you expect the attack rate and the capture rate to be like quite tightly correlated. But the data here only represents attacks made directly at the prey school. And remember, at the beginning of the talk, I mentioned the importance of these isolated sardines. So the logical next step was to look at the isolation rate in these single species and mixed species groups. Well, the isolation rate was actually pretty consistent, independent of whether marlin were hunting alone or hunting alongside mahi-mahi. But, and here's the main result, when mahi are present, the proportion of striped marlin captures that are on these isolates increases significantly. So in summary, these warmer waters likely led to mahi being present in our field site, and the presence of mahi suppressed marlin attacks on the prey school. But the marlin maintained their overall capture rate, and this was because they had a higher proportion of isolated captures in their total ingestions. So what we have here is probably evidence of interference competition, because I think the presence of these large schools of mahi-mahi likely drove away many striped marlin from the aggregations. But in smaller numbers, there's possible evidence of actually a neutralism where the interactions between the species seem to have pretty negligible effects. But I'm curious to figure out why striped marlin were capturing so many isolates. I mean, did they preference these periphery positions to focus on isolates? Or I think more likely, maybe striped marlin were forced a greater distance away from the prey school due to the mahi-mahi attacks. Therefore, the next step will be to do some spatial positioning analysis of the striped marlin with and without mahi present to help us figure out. So we can test for difference in information flow or the rules of interaction, both within the species groups and also between species. And that will be the next thing I do. So overall summary from these case studies, new methods like these drones, for example, but also animal-borne video, biologgers, underwater acoustics, are really going to start helping us understand the functions and mechanisms of these predator aggregations. The nature of interaction seems to be very wide-ranging, so we have to keep working, keep exploring. But we also need more work to have to consider some larger scale data. So for example, the number of hunts individual predators are performing each day. And we need to better integrate the complexities of the hunt. So for example, the movement of the prey school and the prey fragmentation events. Now, lastly, morphology and physiology will be important contributors, of course, but we need a much better effort at integrating these data with attacks and captures and spatial positions. And I'm anticipating that thermal performance, maneuverability, and the interactions of predator morphology with environmental constraints to be really important in determining the species' roles in these feeding aggregations. So I can turn my video back on because that didn't crash. Cool. So yeah, thank you very much. Lots of collaborators from all around the world. It's very multidisciplinary work, but I'd like to just thank 4 people in particular. Jens Krause, who's been a big supporter and contributor to all of this work for many years. Max Litsch, the PhD student I was talking about. this Hector Villalobos, who's one of our collaborators in Mexico, helping us with a lot of the environmental data, and Alicia Burns, who is another postdoc exploring these predator-prey interactions in the open ocean. So thank you very much.
Okay, thank you very much, Matt. That was a very cool talk. Thank you. Yeah, it's very cool how you are using technology to reveal this kind of a strange interaction in the ocean that we didn't know before. Well, right. Okay, people. So keep going with the questions. You know, you can write down your questions in our chat box. I'm happy to introduce our last speaker, Dr. Meredith Palmer. Meredith is at the Center for Biodiversity and Global Change at Yale University. Meredith is a conservation scientist with over 1 decade of experience studying large mammal ecology in African and North American ecosystems. Her current focus lies in integrating in situ and remote biodiversity sensors to develop data-driven strategies for protecting and restoring ecological systems at global scales. And today she's going to talk about ecological and social drivers of group formation in African ungulates. Meredith, thank you very much for being today with us, and please go ahead with your talk. Yeah.
Oh, fantastic. Thank you. All right, can everyone see that?
Yes.
So echoing everyone's thanks to you, Gabriel, and Evan and ATBC for this opportunity. Like Matt, I'm a little worried about my internet connection, so I'm going to go see if I can turn my camera off for now. And I just want to start off by saying that I am presenting on behalf of my collaborators and co-authors. So the true brains behind the research that I'm sharing here are Doctors Julia Saltz and Lydia Boudreau. And so what I'll be sharing are several studies that we've conducted together on the community ecology of African ungulates. So specifically how these species are interacting in shared ecosystems. And it's hard going last, or maybe it's easier going last in a talk like this because some of these ecological concepts have already been really well described by Giselle and Matt. But just underscoring here again that understanding the role of species interactions within communities is a central fundamental focus of ecology. There has been a long history of research on negative interactions, such as competition and some of the interactions that Matt's just presented. But more recently, relatively speaking, there has been a growing interest in these positive interactions where one or both species are benefiting and neither species is harmed. As I think Giselle presented really well, these kinds of interactions can vary over space and time, and as we've heard, depend on environmental conditions. And so the broad challenge here is figuring out where, when, and why positive and negative interactions are likely to occur and what's driving these patterns. So the form of species, interspecies interaction that we're most interested in for this seminar series is mixed species groupings. So defined here as stable or transient associations where individuals of different species are spending more time together. They're together more often than we'd expect by chance. As this series demonstrates quite nicely, this kind of grouping is widespread across animal taxa. And although not all of these associations are strictly quote unquote social, many do involve coordination, alignment, or co-movement in ways that suggest shared ecological incentives across species. Taking perhaps a quick step back to think about this more broadly, the formation of social groups reflects a complex array of costs and benefits, so trade-offs that vary again across different contexts. So compared to solitary individuals, those in groups, whether it be single species group or mixed species group, individuals will experience substantially different levels of predation risk, competition for food and mating opportunities, and access to information. So here we'll be talking about those two classic evolutionary drivers of group formation, foraging and anti-predator trade-offs. And so a lot of these, I think about these in ungulates. They're clearly quite similar in bird mixed species associations as well. But grouping can be selected for example, because it can allow individuals to spend less time vigilant for predators, so allowing more time for other behaviors such as foraging or finding mates. We also have the dilution effect. So where in a group, each individual has a lower chance of being targeted by predators. And also the movement of multiple individuals in large groups can sometimes confuse predators, so lowering the odds of a successful attack. But of course, group living is not a free lunch. There are associated costs of group formation. So larger aggregations of prey may be easier for predators to locate, and individuals within those groups can face higher competition for resources. So that's thinking about the drivers of sociality broadly. These trade-offs can then shift in heterospecific groups compared to conspecific groups. And so here I'll be focusing on mixed species groups that include 2 or more species from similar trophic levels. And their associations, the patterns of these associations offer a window into whether and how different species benefit from grouping together. So mixed species groups provide additional anti-predator benefits in some cases compared to single species associations, such as when the second species is better at detecting predators or uses a different sensory modality. So this can again improve predator detection and reduce vigilance cost. And so we found, for example, that zebra in mixed species groups spend nearly an hour more per day foraging than those in sized single species groups. And this is due to a reduced investment in vigilance behaviors. That being said, some benefits like the predator confusion effect, so a key mechanism by which groups neutralize predator attacks, these might be less effective in mixed species groups due to phenotypic dissimilarity among group members. And predator preferences can also impact certain group members more than others. So this is a classic study in groups of Thompsons and Grant's gazelles. So this is a picture of a Tommy in the big picture and a Grant in the inset. Cheetah attacks are disproportionately directed more towards these smaller Thompson's gazelles, which represents a key benefit of mixed species groups for the Grant's gazelles and a major cost of mixed species group formation for these the Thompson's gazelles. In terms of foraging, mixed species groups can reduce competition if members have partially distinct diets or complementary foraging strategies. And this is actually thought to be a key driver underpinning ecological phenomenon like the Great Migration that happens across the Serengeti Mara ecosystem. And in this interaction in particular, smaller bodied grazers, specifically those Thompson's gazelles from before, they require higher quality food resources. They forage more selectively. And these species benefit from foraging alongside larger, less selective grazers, including wildebeest and zebra, as these heterospecific grazers are exposing fresh plant tissue, so increasing the gazelle's ability to feed on those highest quality bites. But of course, this is a non-exhaustive list of trade-offs, and many aspects of these dynamics have yet to be fully explored. So of this broader question, then, what we wanted to look into were the ecological and social drivers of mixed species groups formation, specifically in aggregations of African ungulates. And to address both of these questions, we tapped into a one-of-a-kind wildlife monitoring network set up in the Serengeti Mara ecosystem, specifically in the core of Serengeti National Park in Tanzania. And just to begin with, the Serengeti is the ideal natural system in which to test the drivers of mixed species group formation. It's home to these hyper-diverse ungulate communities. Many of these species of ungulates have been observed in mixed species groups. The apex predator in this system, so the African lion, feeds predominantly on group living prey rather than on solitary species. Yet prey gregariousness reduces lion hunting success through mechanisms such as decreasing predator search efficiency and improving group defense. On top of this, the ecosystem is strongly seasonal, so food availability changes dramatically throughout the year, altering patterns of competition within and between species. And lastly, the Serengeti is one of the most well-studied terrestrial ecosystems with over half a century of prior research into these species, their interactions, and natural processes across trophic levels. So we focused on 6 grazing species that comprise 85% of the Serengeti lion's diet. Dietary overlap between these focal species ranges from about 25 to 75%, which indicates the potential both for resource competition between some species and facilitation among others. We also have populations of these species. Some of them are resident within the Serengeti year-round, and others migrate following resources across the larger Serengeti Mara ecosystem. But with so many focal species, this begs the question then, how do you study the distribution and interaction between hundreds or thousands of individuals across these multiple species at the spatial and temporal scales necessary to investigate the questions that we're interested in? And our solution to this was to set up a 1,200 kilometer square camera trap survey grid. So 200 camera traps covering an area in the center of Serengeti Park, an area approximately the size of Hong Kong. So these camera traps, I think these are tools most people are familiar with now. These are semi-autonomous remote cameras that capture images of passing wildlife, and they enable us to gather information on entire wildlife communities. So where animals are, when they're active, what they're doing, and most critically for these studies, who they're with. So our camera traps at this point, they've been running for over a decade. And the camera trap study area intersects with areas of long-term lion monitoring, which enables us to map out the spatial distribution of predation risk. And then we compare these two data streams with satellite products of vegetation quality to assess the spatiotemporal variation in resource availability. And a really quick shout out to thank the thousands of citizen scientists who helped us process all of the images produced by our camera traps. I said this project's been running for over a decade, and before AI came to the scene, it could take our research team years to process the images produced in a single year of camera trap monitoring. And so platforms like the Zooniverse and Snapshot Serengeti, which is running to this day, they help us crowdsource the classification process to willing volunteers whose contributions continue to make these large-scale monitoring efforts possible. So going back to the question at hand, we considered any image to contain 2 or more individuals from one of our focal species as a group, and to identify groups that were close together, both in space and time, we organized our sightings into time sites. So that is groups that were photographed in the same location within the same period of the day, so morning, midday, or evening. And we started by examining variation in species interactions along environmental gradients. And the hypothesis that was driving this work is a hypothesis known as the stress gradient hypothesis, which posits that positive interactions between species become more common as environmental stress or consumer pressure increases. So example, an organism faced with a high risk of being eaten may form an association as a defense against their predator. But on the other hand, an organism experiencing environmental stress, such as resource limitations, they could either form associations with other organisms that improve food availability through facilitation, or avoid associations that increase competition. This is a hypothesis that was generated within and has received extensive attention in the plant community ecology, but with only a handful of tests in mobile organisms. And furthermore, which was interesting to me, few empirical studies have examined multiple co-occurring stressors. So both, for example, predation risk and resource availability. So we tested the generalizability of this hypothesis using the occurrence of mixed species groups as a proxy for positive species interactions. And we modeled whether predation risk and food availability predicted the likelihood of mixed species group formation in these Serengeti grazers. So again, if mixed species groups enhance protection from predators, we'd expect them to be more likely to form in increasingly dangerous areas. If the groups facilitate foraging, they should occur more as resource availability decreases, or if competition is at play, occur more as food availability increases. And so using more than 115,000 camera trap observations collected over a period of half a decade, we found that our mixed species groups were indeed more likely to occur in risky areas. So areas that were closer to lion vantage points, which you can see on the bottom of this figure, or in woodland habitat where lions prefer to hunt. They were also more likely to occur during periods of the year when resource levels were high. So these results are then consistent with the interpretation that stress from predation risk may contribute to the formation of mixed species groups, but that competition for resources may prevent their formation when food availability is low. Critically, however, models exploring only ecological factors that determine where and when mixed species groups are observed, these models assume that animals always have the opportunity to form mixed species groups when they need to. And in reality, social opportunities such as weather and who is available in your immediate surroundings to group with, these can vary across space and time. So animals aren't choosing from among all possible groups for who to join, but only among the ones that they encounter. And so as we alluded to before with the gazelle and the cheetah, the costs and benefits of social groups, as well as their ecological and evolutionary impacts, depend on the identities of group members. So this can include age, behavior, or personality of conspecifics, but also all of the traits, species traits that differ among heterospecifics. And so we asked two questions then. First, what mechanisms influence the availability of heterospecific social partners? And so for this question, we modeled the likelihood that each species' local social context, so their time site from before, included at least one potential heterospecific social partner. And we could then ask, when heterospecifics are available, what or how does that shape whether mixed species groups actually form? And so here we modeled the likelihood that each group observed was a mixed species group rather than a single species group or a lone individual. And specifically, we tested whether group type was associated with the number of nearby heterospecifics, either lone individuals or groups. And both of these analyses, as before, accounted for environmental variables like lion predation risk and resource availability. So first, looking at the availability of social partners, we found that ecological drivers, so specifically resource availability, did play a key role in shaping the availability of heterospecifics, so of other species at specific locations and times. For our migratory species, resource availability increased the likelihood that heterospecifics would be present, while the opposite was true for our resident species. So migrants in this system are likely or are tracking seasonal changes in food availability, while our residents are seeking out possibly limited high quality food patches. So this is a coefficient plot of our availability results. I'm sorry, these figures aren't more intuitive, but the different colors here represent different species. And you can see strong effects of NDVI or resource quality on species availability. We also had species availability changing with time of day. So there was an increasing likelihood of heterospecific co-occurrence at midday. So in general, what this is showing us is that the number of conspecific groups at a time site was positively associated with heterospecific presence, indicating that these species tend to co-occur in similar areas of the landscape during similar times of day. But while these ecological variables shaped availability, they did not then predict whether mixed species groups actually formed. So some general patterns that we found here: typically larger groups were more likely to be mixed species. So possibly because individuals prefer to be in larger groups regardless, possibly because mixed species groups may be above a certain threshold, if a certain size are preferred compared to similar sized single species groups or smaller heterospecific associations, or possibly because there just aren't enough conspecifics to go around. Additionally, the availability of heterospecifics at a time site increased, or as that availability increased, so too did the likelihood of mixed species group formation. So this suggests an important role of heterospecific availability in generating these heterospecific interactions. So overall, these patterns suggest that species may prefer to associate with heterospecifics when they're available, or that they simply prefer larger groups of any type over being in these smaller associations. So clearly, landscape level factors are shaping animal distribution in these systems, with resource availability creating the opportunity for mixed species group formation by influencing localized species co-occurrence. However, once animals find themselves together in space and time, social dynamics appear to dominate the small-scale associations and ultimately produce the observations or the observed mix of single and mixed species groups. Our findings demonstrate that not all individuals have the opportunity to form mixed species groups at all times. So limitations on partner availability shaped the propensity for mixed species group formations. And taken together, these results suggest that the ecological and social context, we really need to be integrating this into studies of mixed species groups, which currently tend to assume that such limitations are absent or less important than other factors. Finally, despite the kind of general patterns we've talked about just now, no two of our focal species showed exactly identical patterns across all of the variables that we considered. And this heterogeneity points to the need for more research in this system, as well as the need for continued development of a unified framework for understanding group composition within and among species. And so with that, I'd like to thank my co-authors, the leads again of this publication, Julia and Lydia, the organizations that have made this research possible, and everyone here for their time and interest. And that's all from me. Thank you, guys.
Awesome. Thanks so much, Meredith. I want to thank you and Giselle and Matt for really interesting presentations. And I also want to thank all the people who've been here, listening for about an hour now. So we want to prioritize you guys because you've been here, and we really appreciate that. So what we thought we'd do is we'd start off with some questions from the chat box, and these are kind of specific to each person. So maybe we'll take a few for Giselle since she was first, and then Matt, and then Meredith, and then maybe Gabrielle and I have quite a few questions, other questions that we'd like to ask that are kind of broader term, like, how did you get into all the science and your own personal story and stuff like that? Okay, so let's start with Giselle. And I had to kind of move my cursor here to get to there. So give me a sec, because I know I saw those guys coming in. And I'm going to take, I'd like to call out the participants who have made the questions, but I will probably not say your name right, so apologies there. Bolamvu Franklin Giselle asked you, thank you, dear Giselle, for your presentation. I'd like to know, how do birds choose their leaders, and is the leader freely accepted in the flock? How would you answer?
Okay, so I think that I cannot answer if birds choose their leader, right? Because there are so many preferences associated with some species or with others. I think that it's more what is going on is that leaders have some intrinsic behavior that attract the other ones, right? I don't know, perhaps a leader species, at least in my study area, that I found that leaders are more mobile, more dynamic. Perhaps that movement just caught the attention of other ones that decide to follow the leader. But I don't know if at some point the growth of the flock is just not attracting perhaps other species, right? So perhaps it's not that you have the leader one species will be attracted to the leader, but perhaps other species will be attracted to this species rather than this one that is behaving as a leader. So that will be a really interesting question to ask, but it's really difficult to track the movement of a whole bird community. But yes, that will be, I think that I answered your question. So for one side, they have intrinsic features that attract others, and it's difficult to tease apart who is following who in a mixed flock.
Mm. Yeah, I appreciated those fish, looking down on them. They were like on the water. There's no trees around there. You can tell who's doing what, you know. In birds, we have to, it's very difficult. Okay, so one more question for you, Giselle. It's when you Joe says, hi Giselle, do you know anything about the dynamic between different members in granivorous flocks? Do they also have leaders and followers like the insectivorous ones, or are they more like aggregations?
Well, that's a really interesting question. So especially there are granivorous birds that feeds more strictly on seeds, and there are granivorous birds that can change or they are more flexible, right? Especially with Sporophila, they are a migratory species, so they move to breed in places where there is like a patch of seed, a patch of grassland fructifying and having a lot of seeds, right? And when they are there, they mainly feed on seeds. They do not have all these mixed flock behavior, right? But there are granivorous birds that lives more in environments where they also may find a lot of insects. And what they do is they behave in a similar way, with some species more like warbling finches, perhaps if you know them. Warbling finches start to become have these key roles in mixed species flock. So it depends on the season. For granivorous berries, more marked the seasonality and the behavior they have, and also the composition. So sometimes they will behave like a normal, regular insectivorous bird flocks, but in other moments they mainly switch from eating seeds to eating insects. And when they do that, they behave more like a true mixed flock, or if you want to see. But the Sporophila particularly are not so much of a mixed flock, but they come together. They come together too.
Great. Okay, Gabriel, you want to pick a few from Matt?
Yes, yes, I can do that. So Matt, I have a few questions for you. I'm going to start with one from Meredith Palmer, one of our speakers.
Can I ask it myself?
Oh, yeah, please go ahead.
Sure. Matt, I think I work in the conservation technology space now. And I think the tools you're using to study these interactions are clearly unlocking our capacity to see things we never would have seen before. But I just wonder if you're capturing with these tools all of the predator-prey dynamics, like is it representative of what you think is going on in the larger system? Or do you think that given that you're just seeing things that are happening at the surface, that there could be different factors at play in other parts of the ocean or places that you're not able to monitor with the tools you're using?
Yeah, no, that's a great question. Of course, absolutely not. We're definitely not capturing everything. So we started off with stereo video cameras, and you can see, 10, 20 meters at the surface and a little bit below you. But we know, of course, that the water depth in Baja goes down to 100 meters before it gets to the shelf. And so the sardines, we have some acoustic data showing the sardine schools down at about 100 meters. So we know that they go down there, and they have to come to the surface. And so some people theorize they come up with the tide, with the moon, but I'm quite sure that there's some predator herding to the surface. And so we have so far haven't captured predator-prey interactions with any acoustics in our system. But we've just been piloting the underwater acoustics, and there's other groups around the world who are absolute experts in this, who are recording the interactions between predator and prey in a much larger scale than we are able to do, because they have these long-term permanent observation stations set up. and they will be a combination of sonar with environmental detectors, so things like they can measure upwelling of nutrients, temperature changes, light changing, and they can keep them out there for months at a time and really record the whole system together. So I know we are definitely capturing just what's happening at the surface. And the other weakness that is that when we're filming with the drone, for multiple years now, we've employed the same kind of method where we focus the drone on top of the prey school. And that's sort of the center of attention. So we have a lot of great detail of what's going on at the prey school, when the predators are actually attacking it. But of course, these predators are hunting all day long. And they must be going to multiple prey schools. So we don't know, for example, how many prey schools they're going to in a day. And this is, of course, extremely important if you're going to try and make generalizations about functional benefits. So we've been doing some tagging work, some short-term tag deployments to try and get a better idea of how much energy they're using for different activities while they're searching or actually actively hunting. And then also how many of these bait balls they attack during a day. And then ideally, what we hope, we might observe some of this herding behavior with the tags as well. So to summarize, basically, absolutely not. We are definitely not capturing everything. We're getting a lot of detail about a small part of the hunt. Yeah.
That's so cool, though. I mean, the technology that you're using is how you're pulling all those data streams together is really fascinating.
Okay, great. So I have a question from Giselle. Giselle, would you like to ask Matt directly?
Yes.
Yeah.
I will try to read because I put it there. But at some point when I started reading about mixed flock of birds, one of the mechanisms that researchers used to explain why they come together was, okay, because this is happening on fishes, birds come in a flock together, so to confuse the predator, and the predator won't know which bird to hunt, right? But I discarded that mechanism because birds predators are really focused or target some birds, and so the mechanism is different from fishes, And I was wondering whether, if when a predator come to one of these prey school or herd of fishes, if they eat everything they can eat, or they just eat at some point and go to another one. So the true answer would be, do you think the prey schools are actually benefiting of being in a herd? If the merlins or other predators are eating and everything they can, or they have like a saturation peak and after that they go.
Yeah, that's great. So they do definitely benefit from these cohesive defenses, both by confusing predators, but also through dilution. They can become saturated. So, but the more complicated, I guess, part of your question relates to whether they all get eaten or some get away. Because you would think for this behavior to evolve, some must survive to pass it on. So yeah, sometimes they do escape. So we have seen schools dive down and escape. Sometimes they get brought back up again by the predators, but sometimes we don't see them come back up again. We see this amazing behavior that we haven't published on yet, but is going on in our research group, where the school will cling onto a predator for defense. So actually hide on a large predator and then use them to escape. It's harder for the other predators to attack. And in terms of how much do they eat and do they give up? Yes. So if the attack rate is too slow, so if they're not getting enough, it's obviously all about intake rate more than sort of total amount. The rate is very important. Then yes, sometimes they'll leave the prey school. So in that sea lion example, if the marlin attack rate drops too low, or this also happens if there's too much tourism around, the marlin can't attack, then they will actually leave and probably go search for a different school. So I hope that answers your question. But I was also curious with your system about the cohesion in the flocks? Because I mean, I imagine some under predation threat in these mixed species groups, they probably flock quite cohesively, or maybe not, I'm not sure. But do they have cohesive flocking behavior when they're searching for food, like if they're attacking insect swarms? Or is it sort of this more jumpy, patchy sort of movements, or do they actually synchronize in their flight?
Well, thank you for your answer. It was amazing. So probably it's a combination of yes, it's a benefit because if otherwise it will be a trait that it won't be selected, right? But at the same time, some prey school will be less benefited than others depending on how fast or slow they can cheat the predators, right? Thank you for answering.
Yeah. Also, I mean, sometimes they're so large that they can
Mhm.
survive until the night, right?
Ah,
So
okay.
most of these are visual hunters. So if they can get to the sunset, then they might have survived for the day, and they can
Okay.
wake up and get attacked in the morning. It's not nice to be a sardine, for sure.
Okay.
But then there are also some predators that capitalize on this grouping behavior. So if you think of all of these lunch feeding whales, they actually want the prey to be in a group. They don't want dispersed prey because it's not efficient enough to capture. So they actually capitalize on the prey grouping.
Okay, awesome. So, just quickly to your answer, yes, they have cohesive movement. So you can follow them, but it's not so cohesive as your example, or perhaps in the pictures that Mary did show, it's not like that. So sometimes you see one individual in the canopy and another in the understory, right? And they are moving together. It's like a funnel or tunnel of birds. I will call it in that way. And some of them, sometimes they just remain behind and the flock start to move and the birds try to capture the distance by flying a long way. So it depends. You have like slow and fast foragers, and they try to accompany to match the speed of everybody, but sometimes they just disperse a little bit, they just come together. And recently they have teased apart ant followers from mixed species flocks, because ant follower species that use the army ants, for example, they are called an aggregation because actually birds are there because the food is aggregated there, being visible because the ants are there, right? So they have cohesive, but not so much as in your study caption.
Yeah, no, that's great. It's very cool. And how do you monitor all of the birds at the same time? If they're either in canopy and down below, do you have a team of researchers all following different birds, and you put the data together at the end, or?
No,
So
they're not what you can do. I answer as a birder. Okay.
Yeah,
Go
I'm
ahead.
sure I'm sure in 10 years I'll absolutely be able to do it. Yeah.
see, so I think it's not easy. I usually went with my partner that he's a bird watcher too, and we have learned a lot to be able like fast identifiers, so you can keep the track of all the birds. What I did was like focal observation of behaviors, and I try to see all the birds that are in the flock, right? And after that, I start to do focal observations. So I have, in some cases, I grab 2 individuals in a flock because it's not so easy to follow and record the behavior. And others, I just more individuals or less individual. It depends on my luck. But to track the species that are being part of the flock, if you're a good bird watcher, you can do it fast.
Okay. So experience
Okay.
is very important.
Cool.
Right.
Okay, hey, I want to move on to Meredith. And I'll throw in a question here, Meredith, because you asked something a bit similar to Matt, which is kind of technology, right? So in the camera traps, you mentioned all this work that was done by the citizen scientists to identify animals, right? But now you're moving, it seems, towards automatic attention through AI or stuff like that. So what kind of, can you be missing things there? Like, it's kind of a different, like, how do you, can AI have some biases maybe towards detecting some things and not other things? And I guess more broadly, my question is, where do you see the technology of camera traps going? How is that going to change over time?
Yeah, 100%. And this is something that we have a long history of work in the computer vision and AI for camera trapping space, because I think we're like the first idiots to throw up several hundred camera traps and expect to be able to process all of the data coming in from that project. So back when we started the camera trapping project, AI wasn't the force that it is today. And one of the reasons that we couldn't apply AI to help process the data coming in from our camera trap grid is because we didn't have any of that labeled training data. So we typically train AI. They're the ones we use in computer vision are neural networks that are built to mimic a human brain, and you show them these associations between an image and a classification, a picture of a zebra and the label zebra, and you show the AI enough of these associations and it like figures out what makes a zebra. And you need hundreds of those pairs between image and label in order to train AI, which we did not have until citizen scientists helped us unlock that data and provided us a training set to start building these models and integrating AI into our pipeline. But that being said, AI for camera trap images is still a bit crap. Like, it's not like data that you get from iNaturalist or the kinds of data that might feed like into a kind of like a Google image search algorithm. Camera trap images aren't like nicely framed, beautifully lit pictures of a complete animal. The images that we're getting are at night or during the rain, or the animal is 500 meters away, or you're just seeing an ear or a tail, and it's really, really hard to generalize from those little bits of information. And I think the other big issue we have in using AI for camera trap images is that wildlife communities are long-tailed distributions. So we have a few very, very common species. So we have like wildebeest, zebra, and gazelle. We've got gazillions of them. But then many species that we very, very rarely capture on camera trap. So things like pangolin or zorilla or civets or servals, these animals that aren't as common in the environment. And so we have less training data for those animals, and the AI performs a lot worse on those species. So there's like a lot of interesting things happening in the world of AI now, especially with generative AI that can help overcome those problems. There's new techniques like zero-shot learning, kind of techniques where you can synthesize fake data to train AI on. Like there's some cool stuff going on in that space that's going to make AI for camera trapping better and better and better. But what we do for now, and what I think is a really robust method, is that we try to really rely on the strengths of AI paired with the like highly evolved pattern recognition powers of the human brain. So we use a process called humans in the loop, where we'll use an AI algorithm to take a first pass at whatever is in the picture. AI is very, very good at telling us that there's nothing in a picture. It's very good at telling us if there's a human in a picture or a car. So if we can weed out that data, which tends to be like 50 to 70% of our data set, then we can use humans to come in and either verify whether or not the AI has made a correct guess, or to classify images that the AI is just not very good at classifying at all. So right now we use both in parallel, and that's greatly improved our efficiency at processing these data while giving us the speed at which we need to move through these data sets in order to make closer and closer to real-time research and conservation decision-making. Does that answer your question?
Yeah, great. Yeah, thank you. Now, just to stick to our 2 questions per person, we go back to Giselle. Giselle, you wanna ask your question to Meredith?
Yes. I was curious because, for example, in flocks of birds, they have certain time of the day that they are more active, but you said that during the midday, they seem to be more close together and form this. Do you think that this is related to, I don't know, because they are more exposed at the midday? I was expecting for them to get together at night when more hunters will be there.
Yeah.
I don't know.
This is such a cool question. So my research actually focuses on predator-prey interactions, of which these mixed species group formations are just kind of one behavioral strategy of many that these ungulates use to escape from predators. And I think the reason I love working in these African ecosystems is just how how complex they truly are. I talked about lions in this talk because they're one of the largest predators in the ecosystem. They have the capacity and they do hunt and kill almost all of the herbivore species present in these savannas. But there's not just lions in these ecosystems, right? There's lions, leopards, hyenas, wild dogs, jackals, cheetah, like there's a whole complex, 6, 7 species carnivore community. And so as a prey animal, you're not just trying to avoid predation risk from lions, you're also trying to navigate, this very complicated landscape with predators that are active at different times of day, that are using different parts of the landscape to their advantage. And so there's many more layers of complexity to this problem than we've been able to, we're unpicking it layer by layer, right? And so to speak to your question directly, I think there's two things to mention. One is that lions hunt really well at night. A lot of our carnivores are crepuscular or nocturnal. We know that lions have higher hunting success in the dark than they do in the daytime, or even on nights with a full moon. So I don't think, I think that you've correctly identified a kind of like mismatch in expectations. I don't think the aggregation we see during midday is necessarily against lion predation risks specifically. But that being said, the quality of data that we get at night is much more limited than the kinds of data we get during the day. The camera traps at night, the distance over which they can see, the distance that's illuminated by the camera trap flash is a matter of meters, whereas during the daytime, we could see for kilometers, in an open savanna ecosystem. So I think we're maybe not getting the same quality of data at night, which could contribute to this problem. But I also think there's a lot more forces at play than just risk specifically from lions that are driving these patterns of association during the midday.
Awesome. That is really interesting.
There's a lot going on, as there are in all our systems.
Yes, I know. Depends how deep you go, you start to finding a lot of things going on.
It gets worse the deeper you go.
Hey, Gabriel, do you want to start kind of going to some kind of questions that we can bounce between the speakers?
Sure. Yeah. So we have a few questions. I think we have still a few minutes. So I would like to, I guess it's more in a personal way of you as researchers. So we want to know a little bit more about how did you come out to start with this research? How did you choose these systems and why these topics? Like basically why do you think studying animal aggregations was important or relevant in your careers? So I would like to start with Giselle and then Matt and then Meredith.
Yes. So in my case, it's really weird because at first I started studying anthropology. I think that I really wanted to understand how the world works. But I started to find out that as more as I watch birds, I start to get tangled in them, like start to develop more and more patience. And I just realized how that everything is like connected in the world, one thing to the other. And I really want to understand with my humans limitations how birds see and perceive the environment and the challenges and everything that is going on for them. And I found really fascinating the fact that they can match and be with other such different species between them. So it was like, I don't know, something really fascinating that was going on, and I started to asking questions, and I keep doing it.
Thanks, Giselle. Yeah. So, Matt.
Yeah, so the work I presented today, it kind of progressed quite naturally out of this much longer project on billfish hunting behavior. It's been going on for over a decade through Jens Krause's group and Paolo Dominici. But that research had like very specific questions, as how do they use their bill? How fast do they go? Do they gain any benefits by being group hunters? And so the research is quite serendipitous. I mean, we go out there on the ocean, we bob around on a boat, and we just film what we can, because it's not, we don't know exactly where everything's going to be. And what we sort of noticed is that to answer these specific questions, we had to cut out so much of our video that we had confounding factors in, which were the presence of all these other predators. And then it suddenly became obvious that we were cutting out the vast majority of all the video we were capturing. Because, I mean, if we were just being honest with ourselves and being honest to nature, and just this is simply what was happening. They were hunting in the presence of other animals. These fish aggregations are such a intensely dense source of energy in the system that they're important for so many animals, and they come together, and they no doubt are going to have effects on each other when they're hunting, either positive or negative. And I think because there's such potentially very important resources in the life history of some of these animals, if these are occurring around, if these aggregations are occurring around reproductive events, for example, I thought, yeah, it's very interesting to know who is benefiting and who is not.
I'll close this out then.
Yes, please.
So like I've already said, my background is in predator-prey dynamics, and these hyper diverse, both in terms of prey and predators, savanna ecosystems are just like, oh, they're so deliciously complex in terms of all of these different interactions between species that are going on, and then the trickle-down effects, the impact of these interactions on a broader ecosystem. And so my kind of story is using these camera trap data in order to study predator-prey dynamics. But then because these camera traps, these monitoring tools, they're not just capturing, my lions and my zebra, they're capturing these entire wildlife communities. We've been able to share these data with all kinds of different researchers to address like a whole host of ecological questions. So because of setting up this monitoring project and doing this data sharing, I've been involved in research on everything from like skin disease in giraffes to like the role that termites play in fire dynamics. So we're capturing a lot with these data. And I really do wish that Lydia and Julia, the lead authors on this mixed species grouping paper, were in the call, because they knew about my interest in all of these behavioral strategies that prey are using to avoid predation risk, but they were coming at this from a very experimental, like lab theory based approach. And I hope I'm not misrepresenting here, but I think Julia, the lead author, had been studying sociality in flies and wanted a system where she could, like, fruit flies in petri dishes, and then wanted to see if these same theories were generalizable to natural systems, and we had the right data to address those kinds of questions. So for me, it was like a beautiful pairing of, like, we had data and interest in the broader questions, but the studies were led by these very leading experts in sociality, and it was just like a perfect marriage of data and question in my case.
Great. Thank
Great.
you all for those interesting answers. I hope for our audience, which I hope is composed of a lot of young people, very exciting and makes us all feel that science is so accessible. And here you guys are all pretty young, but you've all really found really exciting discoveries. So I want to thank you, the speakers, and thank the observers. And Gabrielle, you want to finish us out? We're kind of past our 30 minutes for Q&A.
Yeah, so again, we want to thank all our speakers, to Meredith, to Giselle, to Matt for being today with us, to all the participants in our webinar. I think we learned a lot. As usual, we had more questions than answered now, and I think the idea of collaborating between different groups and studying different animal groups in vertebrates has been something really cool, and I think it's going to be interesting to know more about your study in the next decades, right? I think I only want to remind everybody that this webinar is going to be posted on the YouTube channel right after, I guess, the next few days, and it's going to be open for everybody, so people can share it freely with all your
Yeah, and if we
students
didn't
and
get your
labs.
questions in here, send emails to all our speakers, right?
Yeah. So, okay, so thank you very much again, and hope you'll have a very good day or night, right?
Good. Thank you, Alejandra.
Yeah. Alejandra.
Thank you
Thanks,
very much,
everyone.
everyone. It was really great.
Bye.